Today we will discuss about the mechanism of action of enzyme. Now, previously we have studied that what are the factors involved, what are the factors, affecting factors involved in the enzymatic activity, what is their impact and what are their roles. So, in that previous session we have studied that the all chemical reaction is having some energy barriers, right? When reactant molecule interact with each other and forming a product, they are going through a transition state. And this is the transition state. This is the transition state is for the activation barrier. Okay. So the difference between the transitional state and the substrate is called the activation barrier. So here we can see that reactant molecules, suppose A and BC, these are the reactant molecules. If it is reacting with each other, then it is forming a active transition state. So it is forming a complex, that is the enzyme substrate complex, and after that it is going to the product formation. So here this is the activation energy. If enzyme is present, then the activation energy is lower. Here we can see that the purple color now. If enzyme is not present without any type of catalysis, the activation energy will be higher. So this is an uncatalyzed reaction. Here the blue color one and the purple one, this is a catalyzed reaction. So in case of both the reaction, we are seeing the formation of the transition state or the activity energy state. So activation energy is nothing but the transition state. See here, this is a transition state where the enzyme and substrate molecules react first, then after that it's going to formation of a enzyme substrate complex. The minimum amount of energy is required to cross the energy barrier. If if the reaction is catalyzed. So then the enzyme substrate complex is getting activated and form a product with the activation energy. So this is the activation energy, right? This is the activation energy. Here we can see here EA. This is the activation energy. This part, this is the activation energy where the minimum amount of energy is required to cross the energy barrier and gets activated to form a products. Now, when reacting molecule is react more collision will occur, then the reaction will be fast. So this is the main mechanism of action of the enzyme and the substrate molecule. Now here we can see that the enzyme lowers the reaction's activation energy. Without enzyme, suppose without enzyme, this man is taking a big, I think heavy particle. So without any type of enzyme, he needs to move more. The activation energy, it's more, right? But if if enzyme is present here, so activation energy will be less. Net energy is released. So he can easily transport this material from this place to that place. So here, without enzyme, he needs to travel a more, and with enzyme, no need to travel, so the less energy will be spent. So three models are there generally here, based on which the enzymes are working. Three models are first one is lock and key model, second one is induced with model, and the third one is Michaeli-Menten model. What about the lock and key model? Lock and key model, here we can see that the lock and key model is nothing but a model where the theory proposed by German biochemist Emil Fischer, because this model is known as Fischer model also. Fischer template theory or Fischer template model. This fact, the very first model proposed to explain an enzyme catalyzed reaction. So generally this is the first model we can, from where we can understand we'll get the concept of the enzyme catalyzed reaction, how it is reacting with the enzyme. So according to the model, the structure or the conformation of the enzyme is rigid. Here you can see that the structure and conformation of the enzyme is a rigid type. The substrate fits to binding site, that is the active site. Here two active sites are there. We can see here just active sites are there. Here substrate will come and bind to form a enzyme substrate complex. Now after that it will give a product. Two product will be formed. So the active site is a rigid shape, only substrate with the matching type that will fit into the active site, that substrate will come and bind. So this is a substrate is a key and it will fit the lock of the active site. So this is a very older model. Along with that, this model does not give any scope of the flexible nature of the enzyme. So this is a main drawback of that model. This model will not give any type of flexibility of the enzyme. Hence the model is totally fails to explain many facts such as the effect of the modulators. We will learn afterwards what is the role of the modulators, what is the role of the allosteric modulator. So this is a very old model. Just we have to know this is a rigid enzymatic structure they have given. In that structure, one active site is there. One substrate will come and bind with the active site, and after that the product will form. Next is the induced feed model. So induced feed model, here we can tell that this is the active site is flexible. It's not at all rigid. Because here we can see the substrate is structure is different. Substrate structure is different. But here the active site structure is also different. It is not at all fit. By seeing this one, we can understand it is not at all fit with the active site. But active site will change according to the substrate need of the substrate. So active site morphology will change, and then the substrate will come and bind to form a enzyme substrate complex. So the shape adjustment will be there. Right, the shape of the enzyme, active site and substrate adjust to maximize the fit, which improve the catalysis power. So here that is the term here we are using, that is a induced feed. So substrate will induce the enzyme to fit into it. Right, substrate will induce the enzyme to fit into it. Then only it will form a enzyme substrate complex. Right. Next, one more model is there. That is a Michaelis-Menten model. So Michaelis-Menten model, or we can tell that the Michaelis-Menten kinetics also. So this is a very important in the enzyme substrate complex reaction. It is a reaction where the enzyme will react with the substrate and form a enzyme substrate complex. But at the constant enzymatic concentration, the rate of a reaction will increase. See here, here one graph is there. At constant enzymatic concentration, the rate of a reaction means the rate of a reaction means when enzyme will come and bind with the substrate molecule. So this is a rate of reaction will increase, but it will reach a maximum level. That is a maximum activity. That means all enzyme will come and combine with the substrate. So this is the maximum activity level. So this is the maximum activity level. The enzyme will eventually saturate it and giving the maximum activity. Beyond that, we will not get any activity. So this is a maximum activity level, right? So what do you mean by Km? That is a Michaelis constant or Michaelis Menten equation constant. So we are denoting it by a Km. K for constant, M for Michaelis. So this is a Michaelis constant. So this is equal to the substrate concentration of a substrate. We can denote it concentration of a substrate at which the reaction is half of its maximum, right? So this, if I'm putting it in a graph, reaction velocity versus substrate concentration, so we will get this type of sigmoidal curve, right? So here we can see that Km is a half of the substrate concentration. Here the Km, this is the Km value and this is a Vmax by 2, that is the half, and this is the maximum velocity. So it expresses the affinity of the enzyme to the substrate, how the enzyme is binding to the substrate molecule. If low Km, if any reaction is showing low Km, that means what? High affinity of the enzyme to the substrate. As well as in case of high Km means low affinity of the enzyme to the substrate. In that lecture note, I will provide the derivation from where you will understand the equation pattern of the Michaelis-Menten equation or kinetics. So just we have to remember that one, the low Km means high affinity of the enzyme to the substrate, and the high Km means low affinity of the enzyme to the substrate molecule. Next is the temperature. So here also affecting factors is there, lots of affecting factors is there, temperature is there, enzymes are mostly active at an optimum temperature. Previously also I have discussed this one. It will show little bit activity at lower temperature because maximum enzymatic activity it is in optimum temperature, that is the 37 degrees centigrade in case of human physiology. Human physiological reaction or human metabolic reaction is generally occurred in a 37 degrees centigrade temperature. Enzymatic activity will lose when the temperature will increase, that means the denaturation. Enzymatic temperature will lose at the high temperature. When temperature will be higher, the enzymatic activity will be lost. That means the denaturation process. So this is the mechanism of action of the enzyme and the various models I have discussed here. So I'm concluding this part because enzyme portion is almost completed. Just I will provide the lecture note. From that we will understand. If you are having any type of query, just call me. Thank you.